Standard medicine frames aging as an accumulation of cellular damage measured by proxy, cholesterol panels, blood pressure readings, telomere estimates. The clinical consensus has long held that aging at the molecular level is diffuse and fundamentally unmeasurable. But the peer-reviewed data tells a structurally different story. What researchers established in 2013 and a decade of independent replication has since confirmed is that your biological age is not diffuse. It is encoded with mathematical precision at 71 specific locations inside your blood. Locations your physician has never ordered tested. Locations that predict your mortality risk more accurately than any standard clinical biomarker currently in use. This briefing will take you through those 71 sites, what they measure, how they predict, and why the system they track is not a fixed verdict. It is running at a speed that the research has now demonstrated responds to intervention. This is a clinical examination of the 71 CpG sites constituting the Hannum epigenetic clock, the first blood-based molecular map of biological aging published in the peer-reviewed literature. The prevailing clinical framework holds that aging is the cumulative result of random cellular damage, oxidative stress, telomere attrition, accumulated replication errors. The meaningful clinical measures in this model are the downstream consequences of that damage: organ function decline, cardiovascular risk elevation, immune system deterioration. The peer-reviewed data from the past decade documents something structurally different. Aging follows a measurable, reproducible pattern encoded at specific molecular addresses in your genome. A pattern used to predict time to death with statistical confidence that no standard biomarker panel has approached. Before we move into the mechanism, do you currently believe your biological aging trajectory is fixed, or do you believe it responds to what you are doing right now? Hold that question. This briefing speaks directly to it. This will cover two mechanisms. First, the distinction between two fundamentally different readings your biology can produce and why that distinction determines when and how intervention works. Second, the finding that restructured how the most well-capitalized aging research facilities in the world approach the aging process. The data is sourced from peer-reviewed publications in molecular cell, genome biology, and Nature Aging, including the first randomized controlled trial in human history showing that a biological aging biomarker responds to a defined intervention in living humans. Right now, inside every cell in your body, an operational control layer is either maintaining structural integrity or losing it. Think of your DNA as the fixed architecture of a building, load-bearing, foundational, unchangeable. Layered on top of that fixed architecture is a dynamic operational system, a network of molecular switches that determines in real time which genetic programs are running and which remain dormant. In a biologically young system, this operational layer functions with structural precision. In an aging system, the maintenance protocols governing that layer are being systematically skipped. The wrong programs are staying active. The critical repair and regeneration programs are going dark. This is not a future risk, it is a present condition, measurable in any blood sample, compounding in real time. The clinical literature calls this DNA methylation drift. The Hannum clock, published in Molecular Cell in 2013, scanned 485,577 points across the genome, identified over 70,000 that correlated with biological age, and mathematically distilled those to 71 specific sites that together predicted biological age from a single blood sample with a correlation of 0.96 and a mean error of under 5 years. This is an engineering instrument measuring how far the operational drift has progressed in a living system. But here is where the research produced a critical distinction. First-generation clocks like the Hannum model are biological odometers. They measure accumulated position. They do not measure velocity. That required a different instrument. In 2022, researchers at Columbia University published the DunedinPACE clock, a 173-site methylation model designed not to measure where your biology sits, but how fast it is currently moving. A score of 1.0 means your biology is aging at your chronological rate. Above 1.0, the system is accelerating. Below 1.0, the degradation is slowing. The clinical weight of this distinction arrived in 2023. The CALERIE randomized controlled trial studied 220 healthy adults under a 25% caloric restriction protocol for 2 years. Researchers measured epigenetic clocks before and after. The result was the first of its kind in human data. Caloric restriction moved the speedometer. Biological aging pace slowed 2 to 3%. The Odometer-based clocks showed no statistically significant shift. The rate had changed before the accumulated position had time to register it. In independent populations, researchers have associated a 3% slower DunedinPACE with approximately 10 to 15% lower mortality risk, an effect size comparable in magnitude to smoking cessation. The speedometer is the intervention target, but the rate finding only answers half of the structural question, because underneath the rate of aging lies a more fundamental one. Is this a system that can only be slowed, or is it a system that can be reversed entirely? The answer was documented in the same year the clock was first established. The dominant scientific model for aging through the early 2000s held that aging is random damage accumulating over time. Oxidative stress, replication errors, stochastic entropy. In this model, the direction of aging is irreversible because the damage is real and compounding. If this model is correct, the ceiling for any biological intervention is deceleration. You can slow the damage, you cannot undo it. The 2013 Horvath clock study, published simultaneously with the Hannum clock in Genome Biology, contained a result that challenged this model at the structural level. When researchers took adult cells from donors of varying ages and reprogrammed them into induced pluripotent stem cells using Yamanaka factors, the epigenetic clock reset to near zero. A cell drawn from a 70-year-old carrying 7 decades of encoded methylation drift returned to the epigenetic profile of an embryo. The clock did not slow. It did not plateau. It rewound. The molecular layer encoding the aging program is not permanently embedded in the DNA sequence. It is a layer on top of that sequence. And in a controlled laboratory setting, that layer has been overwritten. The 2023 pan-mammalian clock study, published in Nature Aging and drawing on 11,754 methylation profiles across 185 mammalian species and 59 tissue types, provided the structural explanation for why. The CPG sites most sensitive to aging are concentrated at locations governed by a protein complex called polycomb repressive complex 2, the same molecular machinery that controls developmental gene silencing during embryonic formation. In adult tissue, this machinery continues running. What the pan-mammalian data revealed is that the aging program is not random noise accumulating over time. It is a developmental program continuing to execute past its intended phase, a program conserved across 200 million years of mammalian evolution, a program, in other words, with a controller. This is the finding that restructured the research agenda at Altos Labs, the Salk Institute, and Harvard Aging Biology groups, not because it is speculative, because the programmed nature of the epigenetic clock is now documented at the molecular level across the entire mammalian tree of life. The question the field is currently resolving is not whether the program can be modified in a living system. It is the precise parameters under which modification is safe, measurable, and sustainable. Before this briefing, the dominant framework was aging is random damage accumulating until the system fails. No molecular map, no precise measurement, no directional change possible. After this briefing, the framework is structurally different. There is a 71-site molecular instrument reading biological aging position from a blood sample at 96% correlation. A 173-site instrument reading the current rate of aging, with randomized controlled trial evidence that this rate responds to defined intervention, and a body of data showing the molecular layer encoding the aging program has been overwritten in a controlled laboratory setting. Aging is not entropy accumulating passively. It is a program running actively. That distinction changes how you assess every intervention, every biomarker, and every claim about what is or is not biologically possible. The aging you assumed was happening to you is a program running inside you. Programs have code. Code can be modified. The research is no longer asking whether modification is possible. It is working out the parameters.